Hey guys, welcome to Bio 112 Online, your home for biology education. Today's topic, neurons and neuron function. This is going to be a 4-part series. So today we're going to do part 1, resting potential and the Nernst equation. But before we get to the Nernst equation, what we have to go do is think about neurons and neuron structure. You will recall from early in the semester, we talked about the fact that neurons were going to be one of 4 main tissue types. These 4 main tissue types included muscle, connective tissue, epithelial tissue, and neurotissue. Neurons themselves were one component of those neurotissue in amongst other cells such as glia. In terms of the structure of a neuron, they're fundamentally similar to other types of cells. They still have to have a cell membrane. They still have to have organelles and a nucleus. Those organelles and nucleus are found within the cell body of the neuron. Attached to the cell body are going to be additional structures known as dendrites. These relatively short extensions will receive signals from other neurons. The neuron will then take that signal and pass it along to this long filamentous structure, referred to as an axon, starting first at the axon hillock, where it attaches to the cell body, then along the axon, and finally ending at the axon terminus. The axon terminus is the point of the cell that will communicate the signal to the next cell. Now, in addition to that general cell structure, we have to consider other features that are being contributed by other cells. Recall we mentioned those glia. Amongst those glia are going to be cells such as the Schwann cells and the oligodendrocytes. The Schwann cells are found in peripheral nervous system and will be responsible for creating a sheath of protein around the axon. That protein is called myelin and is acting as an insulator to allow the signal to move more efficiently down the axon. The oligodendrocytes do much of the same task in the central nervous system. But we'll get to exactly how they do that in a little bit. Before we do that, let's move on to our discussion of resting potential. In order to understand resting potential, what we have to do is focus now our attention on that cell membrane. So let's zoom in and think about the axon membrane. Along that membrane, we'd find that it's composed of anything like other biological membranes. It has phospholipids, it has proteins. Much like other cells, it also has a charge across the membrane. In the case of neurons, they have a very strong electrical charge, with positively charged ions predominantly found on the outside of the cell and negatively charged ions on the inside of the cell. Now that electrical charge is really important because it's going to be the part that will come through and move the signal along the length of the axon. So how do the cells go through and establish this electrical charge across their membrane? Well, inherently, we first have to think about the composition of ions on both sides of that membrane. Recall that ions such as sodium are going to typically be found more outside the cell than inside the cell, at much higher concentrations. By contrast, potassium will be found more inside the cell relative to outside. The main reason for this is the presence of the sodium-potassium pump. Now you'll recall with the sodium-potassium pump from earlier in the semester again, the sodium-potassium pump will take 3 sodium ions and pump them to the outside of the cell, spending an ATP in the process. They'll then turn around and allow 2 potassiums into the the protein have a conformational change, pumping those to the inside of the cell. Now, inherently, just running the sodium-potassium pump should be enough to create some level of charge across the membrane. This happens because you're moving 3 positively charged ions to one side of the membrane and only 2 back the other direction, leading to a net positive charge outside the cell. However, this charge is not sufficient to carry the heavy electrical charge that's going to be responsible for moving our signal in the neuron. In order to gain that additional electrical charge, we're going to have to go through and use additional ion channels. These additional ion channels are referred to as the leak ion channels. These leak ion channels, from a first cut, seem rather counterintuitive. These are simply open ion channels, just like we've discussed previously in the semester. In that case, they can simply allow ions to move down their concentration gradient. So as we're going through and pumping ions, sodium to the outside of the cell, potassium to the inside of the cell, those leak channels can then go through and allow that sodium and the potassium to then cross back to where they started from. Now, you'd be forgiven if you asked the question, why would a cell want to do this? Why would a cell want to spend copious amounts of ATP to pump cell ions from one side of the membrane only to have them come back the other side? It seems like quite a bit of a waste. But in order to understand it, we have to take into account one more factor. That additional factor is going to be the Nernst equation. In order to address the Nernst equation, we're going to have to come along and move out of the studio and into the blackboard. So let's go now. Okay, so here we are at the blackboard. So what is the Nernst equation? The Nernst equation is trying to predict at what voltage across the membrane will ions stop moving. And by what we mean is this. If you have a biological membrane, like we've been describing all semester, in addition, you have some type of channel protein that will allow one type of ion through. We have a concentration gradient then of ions moving from one side of the membrane to the next due to a high concentration on one side, a low concentration on the other. You say, great, that's just facilitated diffusion. We talked about that at the beginning of the semester. What we can add on to that is this idea that as this ion's moving across, it's also bringing its positive charge with it. If it's not bringing any negative charges, or if there aren't any ion channels for negatively charged ions, then what will start to happen is you'll start to see a buildup of positively charged ions on one side of the membrane, and commensurately negatively charged ions on the opposite side of the membrane. And we say, okay, this is a polarity to our membrane. This is the electric charge that we're talking about. So what does the Nernst equation have to say? Well, basically it's saying this. As soon as you get to this point in the middle, if the charge is built up strong enough, you have a positively charged ion trying to make its way through a membrane. It's moving to a side that's already positive. It's leaving a side that's already negative. It wants to move back into the negative due to its charge effect, but it wants to move forward due to its concentration gradient. Now this creates a bit of a conflict. So what is the ion to do? Well, that ion can move its way across, but at some point it's going to stop. The force of the charge pushing it back where it came from will equal the force of the ions moving down their concentration gradient. When that happens, you reach equilibrium. the Nernst equation tells us about at what charge across the membrane that will happen. So the Nernst equation looks something like this. It is 62 millivolts times log base 10 of the ratio of ion concentrations outside the cell to inside the cell. And what we're saying specifically for an ion, not all ions. So for example, we could consider something like potassium. We said that most cells in the body will come through and have a higher concentration of potassium inside. In the case of neurons, that ends up being about 140 millimolar. On the outside of the cell, we have much lower concentrations of potassium, about 5 millimolar. Okay, so now we have a concentration gradient, which definitely is strong enough to push this ion the other way towards the outside of the cell. But we need a commensurate charge to pull it back against that concentration gradient. The Nernst equation will then say, okay, that charge, E, will be equal to 62 times the log of the concentration difference. We said it's 5 outside the cell, 140 inside the cell. Looking and evaluating this, you're going to say, well, that's going to be well below 1. As a result, when you take the log base 10 of that, it's going to be a negative number. When you go through and do the math, that E ends up being minus 90 millivolts. Or if you measure this point right here on the inside of this membrane, you'd find that this charge was going to be minus 90 millivolts. Great. So what about for sodium? Well, for sodium, you'd see the opposite the case. Because for sodium, we have a high concentration of ions on the outside of the membrane. Repair our membrane from erasing here. We have a high concentration of sodium on the outside, about 150 millimoles. On the inside, we have 15 millimoles. Again, you have the strong concentration gradient. You're going to expect to see the ion want to move across the membrane through facilitated diffusion. But you run into the problem that as it does, it's going to create a positive charge now on the inside of the membrane as it moves across, and a negative charge on the outside. Well, how strong will that charge be if we measure it again right here? Well, we can just plug in our numbers. In this case, if we come along and say, okay, it's 150 divided by 15, that's going to be 10. And of course, the log base 10 of 10 is going to be 1. 62 times 1 is going to be equal to 62 millivolts. Or if you were to measure the voltage right here, you would find it to be positive 62. Let's sum this up. In order to have resting potential, first we have the sodium-potassium pump churning away, moving sodiums out of the cell and potassiums into the cell. As a result, we're creating that concentration gradient. We then have leak channels. They approximate the Nernst equation, which stalls any further net movement of potassium out of the cell. Next, we do have some level of sodium coming into the cell due to the open ion channels, being balanced by a net movement of potassium out. All of this taken together means that the charge across the membrane is something closer to zero than would have been predicted by the Nernst equation with just the potassium channels open, hanging out at somewhere around minus 70 millivolts. At this point, we're set. We're ready to start the next part of sending a signal down the axon. But that's the topic of another video. So, till then, stay safe, stay healthy, and I'll see you later.